Hello, everyone. Welcome. I'm so excited to see so many folks here. We're going to give people a couple minutes to show up. But in the meantime, I want to know where everyone's calling in from. I've got a little poll that's going to be put into the chat. This is also a way for you guys to find the chat function in this webinar because you're welcome to use that throughout this presentation. I'm calling in from Northern California up in Arcata. Oh, we've got people in Stockton, in Davis and Berkeley, Los Angeles. Awesome. Fort Hunter.
Ooh.
All right. We also have this poll here while we're waiting for people to join. We want to know which one you think is cuter, a columbella or a tardigrade. I feel like tardigrades are usually the crowd favorite, but let's check out this group. Oh, we've got more votes for columbella.
Very refined crowd.
Right. Well, thank you all so much for joining. We're so excited to have you here as part of our first quarterly webinar with the California All Taxa Biodiversity Inventory. This initiative works across so many different partners, and a lot of you guys are here in this webinar, so we first want to start off by thanking you all. We're working with universities, museums, researchers, nonprofit organizations, government agencies, all across the state and the country. We're so excited to kick off this 2025, the end of 2025 and the beginning of the 2025 season with this webinar. And with that, I'm going to pass it off to Dan Glusenkamp, who's going to give us a brief introduction about CalATBI and this initiative.
All right. Hi, everyone. Yeah, I caught that use of the word brief, so I'll talk fast and try and get through this. Welcome very much to the meeting. Very excited for the rest of this and the good talks that followed this intro. You know this part. We're working on this because biodiversity is important, and California's got a whole lot of it. So California is one of 36 global biodiversity hotspots. These are parts of planet Earth that have been mapped out, assessed, and we've calculated there's 35 or 36 of them. Together, they're about 3 to 4% of Earth's surface area, land area, and they hold probably something like 70% of the biodiversity. But that's hard to say because most of the biodiversity is still totally unknown, undiscovered, undescribed, especially as you get small and you get into these things that are a little bit hard to tell apart. They look the same or they're just hard to see. And they all look the same because they're invisible, gelatinous little things with incredible incredible genomes and biosynthetic machinery and everything else that is interesting about life, but they're just kind of been too small for us to find with 18th century, 19th century tools. So this is why we're working on this stuff. There are, in addition to the fact that we still haven't discovered most of our biodiversity, potentially something like 90% of the animals are still undescribed. There's that results in big data gaps, the least of which are the data gaps in making our conservation investments and doing conservation planning. So we've got pretty decent data for things with backbones, okay data for plants, but then as you get into the other animals that don't have backbones and they start getting small, we don't have any information at all, essentially, unless it's a single site endemic and it only, it's a butterfly that only exists in that set of dunes. Anything more complicated than that, we don't even have distribution maps for them, much less know what their status is. So there's a big need there. There's also a much bigger need to, to discover what we have, yes, but to back it up. And in the course of discovery, the way we're doing this project, we're backing up specimens and samples of all of that biodiversity today, while we still can, while it's still out there, so that future generations have access to it. to be able to do science, to be able to discover these things, to understand the web of life, to be able to pull out secret recipes for how to live on planet Earth, whatever they're going to need these things for in a generation or two. It is on us to make sure that we have secured them and that they're available in that time. So if we could go to the next slide. All right, cool. So here's what we're doing. We're doing a voucher-based survey, voucher-based inventory of California biodiversity, and it's DNA powered because for a lot of these things, they're very hard to tell apart until you look at their DNA or other really difficult to assess traits, 21st century traits. And we've kind of done as much as we can with 19th century traits like number of digits, and so the focus here is on those things that haven't been done yet. So for insects, potentially something like 75% of our insects haven't been discovered, haven't been described. We don't know if it's 75% because number one, we don't know how many have been described. Number two, we don't know how many are out there. So we're working on it. We're making good progress, as the next speaker, Austin, will tell you. We've then scaled up to include soil biodiversity, which gets vanishingly low rates of discovery. As you get into the soil, you get a lot of really small things that do a lot of really interesting stuff, and we really know very little about them. And soil is important. It's the basis of all of our ecosystems. It's the fundament of carbon storage, all kinds of neat things. And then fungi, similarly. Free living fungi, there might only be 1 million species on planet Earth, but there might be something like 1 billion species that live in other organisms. So fungi is a big goal. We're probably not going to get all of them, but I think we're going to get all of the macrofungi. You're going to hear when FUNDIS and Mycota give their presentations a little bit later today. And then intertidal biodiversity. So not a lot of that representation on this call. That project is just spinning up. It's being hosted now by SCWRP, Southern California Coastal Wetlands Research Program. And it just got funded to be the next add-on in this program series to do a specimen-based DNA-powered inventory of what we've got in this biodiversity hotspot, with the idea that we develop tools, we develop techniques, we figure out what we got, we secure specimens of everything for the future. And once we're done with that, the world can say, wow, California invested a lot of resources in figuring out how to do that. We will do it for the other 34 or 35 biodiversity hotspots on the planet. Back it up. We're talking 4% of the land mass. We can get 70% of the species, which is a pretty good start. So that's the broader goal of the ATBI that's starting with California here. And so I do want to say that we is the really operative thing here. The partnership on this call is just the tip of the iceberg. Tons of organizations, agencies, scientists, communities have been involved in collecting samples, sequencing stuff, processing stuff, going through the difficult work of providing access and permits, all of that kind of stuff. So I do want to say thanks to everyone who's on this call who has worked on this a whole bunch, ranging from adventure scientists to BLM staff, CDFW staff, certainly Stillwater science staff who've been coordinating a ton of this, and everyone else. So if we go to the next slide, I think I'm only halfway through my spiel. So I'll let you do a lot of reading here. So how we're doing this is basically the main deliverable for these projects is to deliver a comprehensive DNA barcode reference library. That means that for every species, we take a voucher and we get some DNA out of it and we get a sequence out of that ties to that voucher, and we can say this is the DNA barcode for that species. And then that lets us reference that species even if we haven't had time to do a thorough species description of it and give it a proper Christian name. We can refer to it and we can go back to that specimen. We're supporting field work to find all of the unexplored diversity, which is a lot. And so field work is probably the main heart of this column, what we're going to be talking about here today, trying to get people out collecting samples. We're also investing in emerging tech to really accelerate the pace and scale of this. Again, the old tools of looking at something really closely don't work. Fortunately, now we're in the 21st century. We've got really good robotics. So we're running these things through robots that can see things in multiple dimensions and a ton, in hyperspectral imaging, and then run that through machine learning and AI to be able to figure out what they're looking at and then shoot those things into the right well of a 96 well plate for next gen sequencing and high scale DNA sequencing of this stuff, and really kind of like really speed up the scale at which we can process samples and figure out what in those samples is important and then sequence it. And then of course, through all of this, we got to do the logistics and the planning. We're generating distribution maps in order to inform our sampling, and then also to use those for planning conservation investments that California and the rest of us are making. And then ultimately to be a baseline of what biodiversity looked like in the 21st century and how it changed as a result of climate change or whatever else, or our conservation actions. And then again, most importantly, to me at least, is preserving specimens, all of this stuff. So we're doing incredible stuff here. It's science, the likes of which God has never seen, but it's nothing compared to what folks 20 years from now will be doing with the material and the information that you all are collecting today. So that's the how. I think I got one more slide here. All right, so here's the pitch on the basic arc. So we're expanding participation and scaling discovery by coming up with really standardized sampling methods and kits. So little things that literally are in a box that you can get in the mail, and you go out and do your sampling and send it back. That needs centralized logistics, maps for planning, centralized permitting. Thank you very much, Stillwater scientists and adventure scientists, for doing that. And then data management to make sure that we're doing this right, and that you individually don't necessarily have to spend 3 years trying to get permits. Instead, you can spend 3 minutes notifying the agency under the existing permits and then go out and do the field sampling. So a standard workflow on this might be something like you contact HOCAN and you request a kit for soil sampling. There's a little bit more in there. You probably need to let Stillwater know which lands you need authorization on. They send you a kit in the mail. It's got some Ziploc baggies and a trowel and some chlorine wipes so that you can sterilize the trowel between samples. You go out, you fill those little Ziploc baggies with soil, put it back in the box, drop it in the mail, it goes to HOCAN. HOCAN gets it in the mail, scans it into the data system, processes it by subsampling your soil sample into a bunch of different pipelines. One of those goes to a flow cytometer for robotic sorting. Another one goes into -80 for long-term storage. Another one goes into -80 for eDNA sequencing, et cetera. And then the soil that you have contributed gets processed in a bunch of different ways. The data all go into a central database. It's shared unless that sample has been flagged as sensitive. It's on tribal land or it's on private land, or in some other way you don't want the data shared publicly right away. And then we move on to the next. And so I want to ask you, as you're seeing all of these talks and you're thinking, wow, that is so cool. I wish I could collect soil and get it run through this process. Please do. Get some of those boxes that you can take out to your field site, talk to other folks in your department or wherever you work, and let them know that they can collect some Ziploc baggies and send them in and get some data back. And then help us engage kind of broader groups that can engage a broader community. So clubs, classes, nonprofits, anyone else who is going out on the land in California to a part that I guarantee has never been assessed for biodiversity the way these folks in the talk later today are going to be doing it. And anyone who's going out to those sites and can scoop up a little bit of soil and mail it back to help push this discovery forward. So that's what we're doing. Hope you enjoy the rest of the talk, the rest of the presentation. Thank you.
Thanks so much. And we'll talk more about how you can get involved at the end of this webinar as well. So don't worry, we'll have more information on that. I would like to introduce our next speaker, Austin Baker. Austin is an entomologist that has been coordinating the insect barcoding effort for the new field collection from the Natural History Museum of Los Angeles County since 2022. He earned his PhD in entomology from the University of California, Riverside, and has spent more than 12 years collecting and documenting California's insect fauna. His work centers on insect systematics, genetics, and biodiversity, bringing together molecular tools and field expertise to better understand patterns of species biodiversity. Thanks so much, Austin. Take it away.
Thank you for the introduction, Colleen. So we have been collecting insects in the field for several years now, sort of just expanding out in the number of places we've gone collecting and just building this enormous data set. And we've done this with certain goals in mind. So we want to maximize the number of species that we're sampling with our DNA barcoding so that we can get as many species sampled as possible to build this reference library. To do that, we've been targeting all the different ecoregions and sampling across all the seasons throughout the state, trying to maximize not only sort of continuous sampling by setting up these long-term malaise traps, but also maximize our sampling across space by just using every other method possible in as many habitats as possible. We have been deploying a number of different traps, active collecting methods, and it's been pretty continuous, and we've built up a data set that we've analyzed through our first 1 million specimen sequence. So it's pretty exciting. Next slide, please. So to date, we have sequenced over 1 million insect specimens. Most of those, about 90%, have been molecularly identified. And so by this, we sequence the barcode sequence, and that gets put into this large database. And where there are gaps or likely speciation events, we get these sort of indications about how many species we actually have sampled now. At this point, it's pretty well identified, at least to the family level, and it's getting continuously improved as more taxonomists visit this data set and improve it. And it's largely composed of Diptera and Hymenoptera, which is not a surprise given the amount of malaise traps we've deployed. But we're getting really good representation of these other groups. We sort of have a comparison in this bottom right of what the number of species we've sequenced versus what is available currently on GBIF. And it's showing we've discovered a huge amount of Diptera and Hymenoptera, which is both probably because there are dark taxa in these groups that have yet to be discovered, but also because they're not totally digitized as of yet. Next slide, please. We've done a really good job at this point of covering as many ecoregions as possible with as many sampling methods. We've sampled every level 3 ecoregion, which is colored on this map here. So you can see sort of the Central Valley there in brown, the Central Foothills in green around that. Each different color corresponds to one of the larger ecoregions. Each of those are built up of a number of smaller ecoregions, which we've sampled almost 90% at this point. So this data set's really rich in terms of its geographic spread. We've also, you can see the different symbols on the map. The red circles represent little snapshot events where we've done a number of different collection methods, and the green triangles are malaise traps, which we set up for months or sometimes even years at a time. So we have these really good long-term sampling data sets as well from this. At this point, where we have gaps is mostly in the Central Valley and the Central Foothills ecoregions. But with our new card nets that are being currently developed, it's pretty exciting that we could potentially get these sites that are more or less converted into agricultural space, still sampled and still integrated into this data set to get some comparative biodiversity information. At this point, we have sequenced over 18,000 different collecting events, but we've collected probably, I mean, this number is a little bit outdated. I would say probably about 4,000 collection events in total. So we have a lot more data that we can actually harvest from this, which is pretty exciting. Next slide, please. When we look at the patterns of biodiversity that we've discovered so far from our first 1 million specimens, what we see are a little bit of a bias in sampling because we've done Southern California really extensively, but I think there is really a pattern there too, where we get in these areas that we've sampled really densely next to these areas that we've also sampled densely, but see a real difference in the number of species. And what we're seeing is a real peak in the Mojave, which even balanced for collections biases and things like that, stands out as extremely diverse. And the Southern California mountains have these really large peaks of insect diversity where we're getting more than 8,000 predicted species within a single square kilometer. That's not all at one time. It could be throughout the year or seasons or as species turn over or change ranges. But it's a huge number of insect species in a small area. And it's pretty exciting to see these numbers once we've actually analyzed all the data. This isn't even a complete data set. We've already filled in many of the gaps with our collecting through 2025, which are not sequenced yet and not added to the full picture. But as those data get added to it, we should really get a really clear picture of what the biodiversity patterns across California look like in a way that could be informative for conservation planning and other sort of larger implications. Next slide, please. And so I just wanted to end on some fun pictures of all of our awesome field experiences. This project has given us the opportunity to cover every inch of California. And I've been to every corner now, and it's just been this amazing experience of seeing all this insect diversity and just the diversity of landscapes and people and experiences. So this is my last slide, and I'm not sure who the next speaker is, but thank you.
Thanks so much, Justin. The next speaker is Holly. Holly Tabers is a project coordinator for the Insect Barcoding Initiative at California Academy of Sciences, leading the sample processing, sequencing, and depositing of thousands of native California insects for online database. I'll get your slides. Thanks, Holly. Oh, you are still muted.
Okay, can you hear me now?
Yes.
Great. Hi, everybody. Yeah, so I am the project coordinator for the CalATBI project here at CalACademy. I work within the CCG team, which is the genomics department. And I understand that a lot of you are field work and collection focused. So we are a little bit of the black sheep here. And we are in pursuit of investigating the genomes of species of interest. And within this project, we're focusing on high throughput. So we're looking at as many specimens, species, family, orders of insects as we can process at one time. And we have a very diverse and pretty large team of volunteers, interns, technicians, and of course, our PI, Athena Lamb. Our former postdoc was Anna Holmquist, who's now working at Fullerton. So everybody's in the lab working every day. Next slide, please. So our role within this project, I'm going to go into that a little bit. Our goal is to create a DNA reference library. We will be uploading that to BOLD and also to ATBI. This project is comprised of about 5 university-based collaborators, 4 organizations within California, several CSUs who host smaller insect collections, and even a few private collectors. We will take in newly collected species from the field, and we also focus on historical specimens. So our collaborators are in charge of plating a 96-well plate full of specimen, and the rest is up to our lab. So that's where we come into play. Next slide. Thank you. Okay, so our job is to extract DNA, amplify, and sequence it. We do this all in-house, and again, upload that for public use in future research. Next slide. So it looks very simple from the previous graphic, but there is a lot of work that goes into that. So the phase 1 of this project was really focused on researching previous protocols, developing and optimizing protocols. The challenging thing here was that a lot of the research was focused on ideal specimen like Lepidoptera that are typically very easy to extract DNA from and sequence, but we're focusing on a very wide variety of as many orders as we can get. So what we found through our research, we have produced a paper here, and we were optimizing certain reagents such as the PEG concentrations, NaCl concentrations, and we really tailored this to the project and created subcategories to achieve the most ideal DNA concentration to obtain those sequences. We did also obtain, oh, sorry, just back one. We utilize high throughput tools such as our BenchSmart here. So this is going to do lab work such as bead cleans on a very massive scale. So instead of working on 1 sample at a time, we're multiplying that by 96 and working on an entire plate at one time, which helps us to achieve efficiency. We're also using multi-channels, so we can work on 8 wells or 12 wells at once as opposed to 1. And next slide, please. Okay, our role here. Essentially, our phase 2 of the process is optimizing our PCR protocols. So we do that to achieve the efficiency within our project. The way we can utilize our collections here at CalACademy is to access our historical collections, which is a huge plus to us. So we're taking advantage of that by having our own entomology department here plate specimens for us. And they have about 18,000 specimens within the collections of representative species. So that is a huge advantage, that we can use our historical collections as well as anything collected newly in the fields. We also do have our resident expert, Athena Lamb, who's very expertised in historical DNA. And the difficult thing with historical DNA is that it tends to become highly fragmented as it gets older. So after about 20 years here, the fragments of DNA shorten, but we can use PCR methods in order to achieve our gene of interest, which is the CO1 barcode here at 658 base pairs. So we're doing what's called a tiling method, and that is targeting smaller fragments, which are still intact, of our insect species and tiling them together. And we do this using a multiplex. So it's several pairs of primers to target different genes of interest within different orders. So it's really applying the same multiplex across all of our insect specimen. So doing this in the most efficient way possible. After that, we sequence using our nanopore P2 Solo, which was generously donated by a donor. So doing what we call an indexing method is another new method that we use that is very uncommon in this kind of lab work. So what we do is we attach unique tags to each plate of specimen. We're able to layer those all together and combine 50 plates onto a single sequencing run. So that brings us to 96 samples on a single sequence run up to almost 5,000 samples. And next slide, please. Okay, and keeping with the trends here, here's our results wrapped. Go ahead and click one more time for me. Thank you. So far we've achieved 21 orders of approximately 31 orders of insects. We sequenced about 426 families. We've completed 8 sequencing runs out of a total of projected 12, and have got our cost down to $2.88, which is great because future groups can continue to do this work on a scale of this size. And we don't have any species details at this moment. I think we will get those as we continue to do uploads to BOLD and to ATBI. So keep posted on that. Thank you so much.
Awesome. Thank you so much. We're going to be switching from talking about insects to talking about the wonderful world of fungi. Our next speaker is Hart. Hart's fresh from the field. The Hart Singer is the sequencing program director for FUNDIS. FUNDIS is the Fungal Diversity Survey. He's an integral component of the California FUNDIS project, having developed the workflow and protocols with input from leading California mycologists. He also manages sequencing for CIB grants and the FUNDIS Rare Fungi Challenge, which engages amateurs to document rare fungi across the U.S. using the iNaturalist platform. Thanks so much, Hart. Take it away.
Thank you so much. We're a little bit further along than some of the CIB projects. We started a little bit after Austin did with insects. Fungi typically don't have great historical collections from California. A lot of the specimens that were collected historically don't have great metadata. They don't have photographs. They don't have DNA sequences. And a lot of them are spread out throughout many different fungaria throughout the United States. A lot of those have really restrictive policies as far as accessing them. So the first goal that we had was to basically establish a baseline for what do we have in the state of California that we can easily access without much of a taxonomic bias. I should mention that this is a partnership, by the way, with Cal State East Bay and my thesis advisor, Dr. Brian Perry, who runs the fungarium there. This phase 1 basically engaged 21 collectors spread throughout the entire state of California to go out where we could get permits for them to collect. And a lot of these are folks that are have experience as amateur mycologists. So they're not just or genus or even species level, and to go to their local areas where they could get permits for and collect just about everything, trying to aim for having, you know, 1 or 2 collections of each taxon. As Dan mentioned earlier, some things are really hard to tell apart, and fungi are one of those things. Molecular barcoding is really the powerful tool that we use to help determine differences between different taxa. So in some cases, we made, you know, maybe more than 2 collections of one thing, but such is the way things work. So we've sampled 56 out of 58 counties. The 2 that we didn't get to are Merced and Sutter. Those are mostly agricultural counties. Sutter is basically a lot of private land in holdings. There's a sort of ancient volcanic mountain range in the middle of California that is very difficult to get access to. But we did really good coverage. We could do better coverage of the desert. However, finding fungi in the desert is not quite as easy as finding insects in the desert. We made just over 12,000 collections, and we were able to obtain over 10,000 DNA barcodes. Our numbers, of course, don't seem to look as impressive as insect numbers, but when one remembers that finding fungi requires perfect weather conditions, they appear not always in the same place. They require a lot of hiking. Sometimes you can go out to the field and expect great collecting and end up with nothing. So to put that in perspective, out of these over 10,000 barcodes, we collected 4,089 unique taxa. And this is, by the way, just the phase 1 data. We haven't delved into phase 2 yet, which is ongoing. Out of those 4,089 unique taxa, about 2,500 of them, as defined by their DNA barcode, do not match any known species. The caveat being that we don't have a lot of sequences for the holotype collections. As I mentioned, they're difficult to get access to. Stephen Russell will probably talk a little bit more about that. Next slide, please. So to establish this baseline, we developed a field to fungarium pipeline. So it's kind of end-to-end complete solution. We would send these amateur mycologists to the field equipped with smartphones and using the iNaturalist platform. We shopped around with a bunch of different companies like Survey123 and FieldNote, and none of them really had the framework to do fungal surveying. iNaturalist is a free tool, so we don't have to pay anything for it. And it has an exceptionally large community, especially in mycology. So we figured this would be a great platform for us to use, and it worked out really well. So folks would go collect mushrooms, take pictures, record the GPS coordinates, and also take down a bunch of really important metadata, including identifying plant species nearby, and any other sort of physical notes. They often carried UV flashlights and record UV reactions, KOH reactions. It's a sort of chemical reaction that occurs on fungi. It'll change color, and then upload all that data to iNaturalist. Next slide, please. So iNaturalist essentially was our data workbench. It allowed multiple people from different specialties to sort of add data to these records. And then because iNaturalist has an API, you can export all of this into whatever format you'd like and upload it to public databases. So the great thing about this is that it's instantly made public. So the second the photograph is uploaded to iNaturalist, people in the mycology community can begin adding their identifications to it, arguing over what it might or might not be. And researchers across the globe are looking at this, waiting for us to upload the sequence data. Also, there's a direct pipeline from iNaturalist to GBIF and NatureServe. So there's no need to create a specific export for them. It's already there, ready for them to be used. And it's user-friendly and widely adopted by the public. There's this robust community. Let's go to the next slide, please. So I'll show the anatomy of an iNat observation. And so the first thing that you see is going to be a photo. We instructed our collectors to take really descriptive photos showing different angles, pictures of the specimen, different conditions. And then in the field, you can write your notes directly on the phone, which will show up in the sort of notes field. And then you have a map with GPS coordinates. Next slide, please. Further down the observation is the sort of key component, which are the observation fields. So as the specimen goes through different stages of processing, different observation fields can be added to this. And again, these are exportable via API, so you can immediately change this into whatever database format you'd like. So you can see on the left, we've got associated species with names lookup. So you can look up all the different plant species nearby. The number of reads used to generate the DNA sequence, the DNA barcode itself, where it's stored, our tag numbers, and then we also include provisional taxonomy. And I'll get into that in a second. Then we also include links to BLAST results, so you can see a permanent record of the interpretation that was involved in determining a species name, as well as the raw DNA sequencing data. And that's hosted on Stephen Russell's website, MycoMap. He'll probably talk about that a bit. And then a link to the specimen location on MycoPortal. And again, all these have been now uploaded to NCBI GenBank and to Barcode of Life Data Systems, BOLD. Next slide, please. So we use the ONT MinION platform, and thank you so much, Holly, for describing the multiplexing, because I wasn't going to go into too much detail. So now folks have a good idea of how that works on a very granular scale. Originally, we started out using Sanger sequencing. We would send our specimens to Barcode of Life Data Systems partner, University of Guelph, where they charged us an arm and a leg to do DNA extraction and Sanger sequencing. And Sanger sequencing was once called the gold standard. I would argue it's no longer, it's mainly the silver standard at this point. I think next generation sequencing would take that gold prize. And that's because you can resolve things that often don't work with Sanger sequencing because there are multiple taxa involved, or because there are multiple variants within the DNA barcode that you wouldn't catch with Sanger sequencing. We're able to do this in-house for 1/3 of the cost that we were spending on BOLD sequencing at the University of Guelph. And we use the ONT MinION platform using the Flongle adapter. So relatively low throughput, but we can still manage to do up to 960 specimens on one sequencing run using the multiplexing techniques that Holly was explaining. We can't use Illumina to get the best taxonomic resolution because Illumina will max out at about 300 base pairs, and our fungal ITS sequence is about 700, so similar to the COI locus. And in order to make the error-prone nanopore sequencing accurate, we use a consensus building process that essentially will take hundreds of reads and use that to polish out any sequencing errors that would occur in individual reads. So I know that's a major concern of people when they see nanopore. They say, "Oh, well, how do you deal with the error rate?" And so we have a very robust pipeline. We actually looked at 700 sequences generated with both Sanger sequencing and nanopore technology before making the jump to this. Next slide, please. So then after the specimens are sequenced, they are then stored directly in the fungarium at Cal State East Bay, where they can be immediately accessible to researchers. Because our operation is based at Cal State East Bay, we can utilize student labor and teach students how to do fungarium work and also teach them how to do the lab work using the nanopore platform. It's a central location within California, so it's, nobody has to go all the way up to Arcata or all the way down to LA to grab the specimens. They're right there in the middle. Next slide, please. So we've now begun phase 2 at the beginning of 2025. And this is to basically, once we've digested the data we got from phase 1, now where do we get all the stuff that we missed in the category of macro fungi? So these are fungi that you can see with the naked eye. And that definition is different depending on who you're talking to. But Phase 1 predominantly got sequences from Mycota, which are the sort of mushrooms that have gills that you would see, really obvious and visible. Our new focus is on Mycota, which is actually the largest phylum of fungi with, potentially millions of species, including the ones that you can't see with the naked eye. But a lot of these have very specific plant associations. So certain plants will have endophytes or pathogens that only occur on that plant species. And California, being the most botanically rich state in the country, gives us a great strategy for trying to find some of these rare fungi. So we sent collectors to very rare places in California. So one of these is the cedars. So that's the picture you see right here. The cedars is just as the crow flies, like 20 miles from my house in Sonoma County, but it takes 3 hours of driving on really rough roads to get out there. It's home to serpentine endemic plants such as the Sargent cypress, and from there we isolated 25 taxa that have never been collected anywhere else in California to this day. So pretty cool. We also just got back from Big Sur, where we did a deep dive into those habitats, including the rare Santa Lucia fir. It's the rarest fir species in the world, and it only occurs in the mountains of Big Sur. Next slide, please. And then sort of to finish things up, we thought, well, how can we make our efforts more useful for the rest of CIB? One of these ideas is oftentimes when we collect mushrooms, we find ones that are in poor condition. They're filled with insect larvae. They're gross. We don't want anything to do with them. But that, becomes a potential source of novel species of arthropods. So not just insects, but also nematodes. Those aren't even arthropods. Nematodes, worms, and collembola, which are these weird six-legged arthropods that kind of resemble insects, but are extremely diverse, extremely rich, and very ecologically tied to fungi in particular. So to get those, we'll take these kind of less than ideal specimens and put them in a Berlesi funnel, which is a screen under which there's a tube of ethanol, and then we apply a heat lamp, and that heat will dry out the mushroom and also cause the various different invertebrates in there to fall into the ethanol. And we found a lot of interesting stuff in those. Those have all been sent so far to Biorepository, where there will be analyzed using the COPAS machine, which you're going to hear about in a bit. We're also collecting soil associated with these mushrooms. And now that CIB has a lot of soil collecting permits, we are trying to build just general soil collecting into our protocol. And thank you so much, Stillwater, for doing that. As anyone who's tried to get permits to do anything in California will tell you, it is a lot of red tape involved. So there's so much more to talk about, but there's so little time. So Stephen Russell's going to fill in a lot of the gaps that I glossed over. Thank you all so much.
Thanks, Hart. We're going to keep talking about fungi. Our next speaker, like Hart said, is Stephen Russell. Stephen Russell is a mycologist and community scientist organizer focused on large-scale fungal biodiversity and DNA barcoding. He founded and directs Mycota Lab, a nonprofit molecular biology lab in Fungarian, dedicated to documenting macrofungi across North America through partnerships with local clubs, herbaria, and individual collectors. Russell has helped coordinate continent-wide mycoblitzes efforts that have overseen the generation of tens of thousands of fungal DNA barcodes that link field observations, herbarium species, and environmental sequencing data. His work sits at the intersection of taxonomy, conservation, and bioinformatics, building the data infrastructure needed to understand fungal diversity and make it usable for research, environmental monitoring, and future ecological tools. Thanks, Stephen. Get your slide up.
Yeah, thanks a lot, Colleen, for giving me a chance to speak for a few minutes about some of our projects that are related to CIB. And so you can really kind of look at our projects as having two separate arms. One is specimen-based and one is soil-based. Our specimen-based projects involve modern collections as well as historic collections. And our soil-based projects involve cultures and eDNA, and I'll talk through each of those briefly. Next slide. So our lab, as Colleen mentioned in the intro, we're really interested in community science at large scales. And one of our primary initiatives is something that we've been calling the MycoMap Network. And on the image at the right, for each of the states that are colored, citizen scientists from all of those areas can send an unlimited number of specimens from anywhere in that state to our lab for free DNA sequencing. And California, the way our efforts of modern collections differ from what Hart talked about is FUNDIS really focuses on skilled and targeted collectors, and we have more of a citizen science first approach to our modern collections. Overall, for the continent, we're doing probably at this point, this year will probably be over 30,000 specimens a year. Up to this point for our CIB project, we've been able to successfully barcode over 6,000 collections. And I would mention that this is this particular project, even though our funding period's ended for CIB, we are going to continue having this open call for California collections of macrofungi. And so if you're looking in, mycomapca.org is the website you could go to if you're interested in helping to continue this ongoing project. Our process is relatively straightforward. If you could hit the next slide. As Hart talked about, we document everything on iNaturalist. So our protocols, we try to keep our protocols as simple as possible. That way to encourage as broad a participation as possible and to leave it as easy on collectors as we can. And so the protocols for citizen science collections ongoing are really just document the mushroom on iNaturalist, dry out the specimen and print a label and mail it to us for DNA sequencing. And so we try to keep that process as simple as possible. And we're going to be continuing this indefinitely for California as a part of our broader initiatives. mycomapca.org if you're interested. Next slide. So that's really was our focus on modern collections. We also had a focus on museum collections, fungarium collections, or historic collections, as some people might call them. And as a part of this project, as was mentioned, I'm the director of Mycota lab. We're actually based right outside of Ann Arbor, Michigan. And one of the largest fungaria collections and most important fungaria collections in the country is actually located at the University of Michigan. And so what we did for our initial museum project was go to the University of Michigan and pull out all the California collections that were housed at the University of Michigan. And so some images of the process are found below. One of the toughest challenges of a museum project for macrofungi is actually just getting the specimens out of the herbarium itself. And so typically you have to open a cabinet, then you have to pull out a box, open that box. Within that box, there's a whole bunch of smaller boxes. And so just the process of getting the specimens out is often one of the most challenging and labor-intensive aspects of an herbarium project involving macrofungi. Some examples to the right of what that might look like in practice, just stacks and stacks of little boxes that you have to scan in, open one at a time, pull out tissue. We actually pull the tissue in duplicate as a part of our protocols, which I'll talk more about in a second. Next slide, please. So on the last slide, it was mentioned that our University of Michigan, California project actually ended up being, it currently would rank as the largest and most successful fungarium sequencing barcoding project that's occurred to date. Most of the specimens were in the 1950 to 1980 range, averaging about 75 years old. So they were really, older collections. Overall, we had about a 70% success rate for those specimens. One of the ways that we've been able to get that success rate up so high is that we spent quite a long time trying many different protocols, trying many different approaches, and it's led to some really good results. Just to put it into perspective, if you were to look at the next largest fungarium barcoding project that would have been done with Sanger, it was the same number of specimens, right around 6,000 specimens, and they only had about a 20% success rate for specimens of a similar age. And so there's a pretty large discrepancy between the types of success that you can expect with Sanger sequencing and the types of success that you can expect with next-gen methodologies. We use a nanopore sequencing as well, just like the other presenters. But we've been able to really develop and hone our protocols over time to give us some really decent success rates at really unprecedented scales. Next slide, please. We do have one more project that is pending on the fungarium side. We've been able to collect tissue for 17,000 specimens at the San Francisco State Herbarium earlier this year. And so the sequencing is still pending on that one. And we're really looking forward to what that's going to tell us. 17,000, if you think about 6,000 was our last project, and that's currently the largest fungarium project. This one more than doubles that previous effort. Next slide. So we talked about modern collections and historic collections on the specimen side. Now if we start looking at the soil side, we did have a project that we're finishing up at this moment in time involving fungal culturing from soil samples. And so this is a project that has about 2,000 fungal cultures that are going to be sent to the CIB's biorepository. We're currently finishing up that project this month, and so the shipment won't be far behind that. But just as with our other projects, essentially what we do is get the vouchered specimen, we get an isolate, as you might see in the middle. The plates on the outside are not isolates. They're some of the initial plates to where we plate out the soil and try to get as much biodiversity. We use a number of different media. We use a number of different incubation temperatures in order to try to get as much biodiversity coming out of those soil samples as possible. Then we would transfer each of those colonies to an individual plate, one of the smaller plates that you would see in the middle. And those smaller plates are what would actually end up being the vouchered specimen for this. And we would end up barcoding it just like the specimens. And so that's the first part of our soil sampling efforts. If you hit the next slide. And the second part of our soil sampling efforts are environmental DNA. And this is something, it's actually been one of the most challenging components of everything I've talked about, but I think longer term, it's going to be the most interesting of anything that we did as a part of CIB. One of the reasons for that is we've developed a number of new molecular methods, a number of new bioinformatic methods, a number of new analytical pipelines for this process. So one of the ways that we've been able to scale our macrofungal sequencing efforts, our modern collections, our community science efforts, one of the ways we've been able to scale that across the continent is not by getting more funding or it's been by becoming more efficient in our processes and methods. And so when we were doing Sanger sequencing, we were probably paying 10x as much per specimen when we started the MycoMap network projects, where people can send in mushrooms. And we've been able, as a part of the methodological development on the specimen side, we've been able to drop the cost per specimen by 10x, but then we've also been able to have about 10x more throughput. And so we kind of look at it as like 100x efficiency gain. And that's really what's allowed us to scale those specimen-based projects. What's really needed for environmental DNA is something similar. Like 100, and not just a small efficiency gain, something like 100x efficiency gain in terms of the cost per sample to get it processed, all the way from sample collection to bioinformatic outputs. And that's really what our focus is on eDNA as well, is to fundamentally change that cost curve of what's possible, because we want to scale just like with the macrofungal specimens. We really want to have continental scale soil projects, and that's kind of the next evolution for our lab, is really to start to scale eDNA in the same way that we've been able to scale specimen-based collections. And from our perspective, the only way to do that is to fundamentally change that cost structure, and we think we're there for that. And so lots more to come in this area soon. And I think that wraps up my component. So thank you all for your consideration.
Awesome. Thank you so much. Our next presentation is talking about our soil biodiversity. And unfortunately, the SoilTech team couldn't join us at the 11th hour, but Dan is stepping up and going to present on their slide. So thank you so much, Dan. I have a quick introduction for the SoilTech team. It was started by two nematologists searching for a better way to count and identify nematodes. The result was this smart soil organism detector, an instrument that can extract, count, identify, and sort organisms from the soil using AI. Dr. Dennis Willett is a faculty at North Carolina State University, where he focuses on applied AI. He is one of the founders of SoilTech, and the other one is Dr. Camila Figueres, who is associate professor at University of North Carolina, Asheville, where her lab focuses on quantitative approaches to soil biodiversity and biological control. So thanks so much, Dan, for stepping up and presenting on slides that are not yours. We super appreciate it.
Yeah, definitely my pleasure. I think this is just some of the coolest stuff.
Hey, sorry.
Oh, you are
I'm
here.
here.
Oh,
Good.
awesome. Thank you so much, Camilla. We made it. Perfect. We've got the
Yes.
expert in the room.
So thank you so much for the introduction. And I appreciate the opportunity to talk a little bit about SoilTech and what we are doing. Unfortunately, Dennis is not here. I will do my best to share what we have been doing so far. Next slide, please. So I would like to start with how this idea comes from, right? Both of us, Dennis and I, we are fascinating with soil and the amount of organisms that we can find there and how hard it is to explore that. So the idea and figure out a good structure to explore them and learn more about the different organisms that we can find on soil makes us create the smart sod that is basically the adaptation on a flow cytometer specific for our needs. So this graphic, for example, shows is not our paper, but this shows how much diversity you can see in just a square meter of soil, for example. This is beautiful, a lot of organisms, everything that we need, especially for this project, right? Next slide, please. But this is hard to study. It's very hard to find, it's very hard to isolate, and it's very hard to identify those organisms. So the idea, it's trying to facilitate that. It's, next slide, please. Is then when we start with the idea with the smart sod. So the smart sod, it's the smart soil organism detector. It's basically a fancy funnel that is gonna look everything that it's mixed with soil and separate by the difference between those organisms. So if you see that we collect the soil samples, we mix in the water, and we go through this big tube that has lasers showing all over the place, reflecting, and collect specific measurements on those organisms. And after that, they separate everything that is different in different 9-6 well plates. Next slide, please. This is a picture showing an example of 9-6 well plates. And in each plate, we can separate one organism per well plate. That is the case for this project, or we can put multiple organisms in each well plate. We can decide what we want to do. And so each well plate, we have a unique organism. Next slide, please. And if you highlight this one well, you're gonna see the organism as a picture, right? That is super cool. Sometimes we can use for identification, but it's not what we are focused on doing the specific separation on it. So together with each individual organisms, we have a picture. Next slide, please. We have also a specific profile, and this profile is what we use to separate and differentiate each organism that is present on this group. So this is like different lines with different colors. It's going to show different reactions, and this reaction could be in different ways. Could be the color, could be the proteins, could be the formation of the cuticle. Different compounds that forms that organism, it's going to react in different ways with the lasers. Next slide, please. So what we have been doing, each organism, we can see on the far right here, we have all the images, right? And those images are great for us to have a big picture what it is, but we can separate by specific species just looking those images, and it's a super high resolution camera. But if you look in your left side, you have the specific profiles, the combination of the lasers. And this combination of lasers create a unique fingerprint for each one of the organisms. And when we create these fingerprints, this is what we use to separate and differentiate each organism from each other and separate those in the wells. Next slide. So this is a graphic showing some examples how the machine thinks. It's my way to think about this. So basically, we can separate big groups. For example, Arachida, this is in purple, separating for Collembola. You guys can see that spatial-wise on the figure, it's completely far away, completely different, right? And we have some groups that is more similar, like all the nematodes. We have them more near each other, but they still have specific groups separate on the dimensions. And this is what we use the machine learning to separate and differentiate each one of those groups. And we start broad and can go more specific by species. And in some case, in nematodes, that is our start point. We can go in strains, that those are nematodes, the same species that we can find in different locations, for example. Next slide, please. So the first thing we want to make sure what we are doing and the way that we are separating them, it's matching specific with the genetics, right? If this matches. To do this, we can link, we did some testing and do some genetics to compare the results. So we was able to get the digital fingerprints explaining 91% of the observed variation on barcoding regions. That was pretty good separation by that. So basically we create those plates, we send for genetics, and when we get back the results, we was able to label those with the barcoding. Next slide, please. So what we try to do with this, right? We want to do genetics. Everyone is doing genetics. So the idea with that, it's trying to save time and save money, basically. And the way that we are doing this, we don't want to send everything for genetics, because the instrument now, what they are doing, the instrument is doing is collecting everything that is organism-wise, that is different than soil, and plate those on 96 well plates. But a lot of those things, as we can see on the figures before, they are already together and have common characteristics. So instead, get a bunch of the same things to genetics. We decide to do an approach when we can create these regions and select the ones that we think it's something completely new, completely different. Or when we have closed this parts here, I don't know if you guys can see my cursor, we have some more strong color here. We have more samples, the same similarities. And when you have some separation in light colors, it's because are things that it's a little bit different than the other ones. So our goal is using this approach to select the things that needs to go to genetics that we know almost 100% that will be a new species or will be a new thing that we didn't went through before. So selecting this, we can reduce the amount of samples that goes for genetics, right? And be able to correlate back with the other samples that we have saved for doing this. So next slide, please. The way that we are doing, we basically start running samples for sure with the instrument working the way that we want in July. And until now, we was able to run more than 500 samples at this point. and separate everything soil organism wise that was there in 96 well plates that you can see that is ready to do genetics. Those are around 7,000 different organisms that will be there on this, and use the approach that we was trying to do in organizing the groups. We was able to prioritize the species that we think it's something unique, and this reduced the amount of samples that we can start with the genetics in 1,200 priority species to go. This, we very low guessing here, will be around 200 new species that will be discovered just doing this approach. That it's the goal with our part of the project, is figure out new species as soon as possible. So together with this project, please, next slide. We was able to do something that we was super excited to do this for years and never had the chance to explore what we can find on the big trees, right? And the unique ecosystems that we can have in each one. So through this project, thank you, Dr. Silet, for provide the samples on this one. And I hope I will be able to do some rappel soon with you. So this is something that will be super cool to be able to do it. We was able to see the unique organisms that exist on the top of the trees that nobody else was able to explore at this point. And we have not just images, but specific fingerprints for each one of those. So we are very excited to be able to explore this and get more information from whatever we're going to be able to explore. Next slide, please. Sorry.
Oh, no, thank you so much. I super appreciate it. It's very exciting work coming out of the SoilTech team and with the rest of our group. Thank you so much to our speakers. I wanted to finish this up by telling you that we've collected thousands of samples, and have you seen, we've discovered tens of thousands of organisms, but we are just scratching the surface when it comes to California. Like I said, it's a biodiverse hotspot, and we have so much more to do. So with that is a call to all of you guys. A lot of you have helped collect samples, and we need to collect more samples. CalATBI is putting on a couple different bioblitzes this year, including this first quarter, a bioblitz in the greater Mojave area, as well as calling to all students to come to California to help collect insects in our statewide student insect collection bioblitz. We're also continuing doing collection rescue. We have all this information up on our website, www.calatbi.org. We're also collecting samples all over the state year-round. Our field efforts start January 1st and end December 31st. So if you haven't already, we really encourage you guys to sign up to be a CalATBI partner if you're interested in collecting samples, and we can send you all the equipment you need and all the information you need to do that. So the last thing I want to do, again, is a huge thank you to everyone. Thank you to our speakers. Thank you to the people on this call. Thank you to the folks who are here, who helped with collections, with processing, with permitting. We really appreciate it. And we do have some extra time on this webinar, and I kind of want to open it up to all the folks on the call who have any questions. Feel free to put something in the chat, or if you'd like, you can raise your hand and I can unmute you as well. So I'll just open it up to the group for any type of questions. Nobody?
One question in the chat.
One question in the chat.
So there's a question in the chat. Can people get involved as an individual?
Yes, absolutely. Scan that barcode, go to our website, sign up. We will contact you and get you all the equipment you need.
If you find mushrooms, you can send them to Steve Russell.
Thanks, Art. I was going to chime in on that one.
Yes, I guess our sign-up sheet is focused on insects, soil biodiversity, but if you're interested in fungi, just put that in the form and we'll connect you to the correct people. We've got a question from Nicole. Can microbes currently in cultures be sequenced too in this effort? Dan, can you answer that?
Sorry, Stephen, were you answering? Not completely clear what you mean, but yes, the answer to most questions in this project is yes. At this point, Paul Carini at University of Arizona is sequencing microbes as part of, taking the soil samples that are submitted and then culturing them for the unculturable bacteria, the ones that no one's ever been able to get out of samples, and then grow them up and freeze dry them after sequencing. So I would probably connect you with Paul if you've got, if you've got some stuff that you want to sequence. There's other pipelines as well, and it kind of depends on what you've got and how many. Do you have a freezer full of plates? In which case we can really mobilize to make the most of that. If it's something that you've got in your fridge left over from some bad cheese, maybe it's a different scene.
Awesome. Thanks, Dan. The next question is from Rose. When groups collect samples for this project, are the results shared with the collectors? And yes, absolutely. We're setting up that platform. It's going to look a little bit different for each sample that you're collecting, but absolutely all the results will get shared back to the people collecting it. Again, it might not be within a very short time frame because a lot of stuff takes time, but once we have everything and all our frameworks set up and going, it might be relatively quick. We've got a question about the student insect collection project. I'll just go over real quickly, but I really hope you reach out to us, go to our website and sign up. But essentially, we want to support you in getting student groups, entomology groups, any enthusiastic insect type people groups to California to help collect insects. And we can support in a variety of different ways, including travel stipends, potential permitting help, prioritization, travel logistics, etc. So contact us. The information's on the website. And I think actually Christina Lincoln might have put that form in the chat as well, but that BioBlitz as well as the Mojave area BioBlitz sign-up sheets are on our website.
And Kyle, you've got kind of another element of your question is around, an old collection that's gathering dust somewhere. So there's another ongoing project to rescue collections that are at danger of loss or damage. And so I'll put that in the chat here. So if you have material, especially samples, which, most museums just don't have the capacity, the space to store samples, boxes full of cups, full of stuff that's unsorted or unidentified. We want to secure those for the future because technology is making it much easier to access those and figure out what's in them. And we want to make sure that stuff is available for future generations. So if you all check the chat, if you know of a lab that's, the PI is retired and they need to move the stuff on, anything else, that's an operation that will help find a home for it. If it's quality material that's been identified, then we'll try and make sure that it goes into a durable collection in one of the natural history museums. If it's, a pallet of banker boxes of sample cups, then we'll get it into long-term storage. So collection rescue, everybody.
There was also a question on fungal diversity, if they can collect mycelium only. Historically, the answer to that question would have been no, because most herbaria would not accept collections of mycelia only. But for this project, we love sequencing collections of just mycelium. And so the main challenge there is ensuring it gets to the lab in a condition to where we can find it appropriately. So if you pick up a bit of mycelium from the soil or something like that, and then it gets smashed up all in the soil, that becomes more challenging to sequence than if it's isolated and dried external from the substrate. And so it really kind of depends on the collection itself as to how easy it's going to be, but we certainly are interested in collections of just mycelium.
We also have one last question from Laura, who's asking if we're working on cataloging existing collections. Dan, do you want to take that one?
Cataloging, what was that?
Existing collections.
Yeah, yes, we are peripherally, and so please get in touch with Collection Rescue if you I'd like to learn more about what you're talking about, and I'm sure that crew would as well. We don't have a formal catalog, but we are working with several other catalogs, lists, Excel spreadsheets. At this point, there's something like 280 collections that are recognized in museums in California. But there's a number more in research facilities, field stations, local visitor centers, stuff like that. So we want to find them all. Everything that we're doing is we're tracking it, and ultimately we'll be making it publicly available. So any info that you are able to provide to that team will help them in their mission, and also will not be lost. Will be added to the corpus of information that we all need.
Awesome. And with that, we're at time. So thank you so much. Thanks everyone for joining and participating. Again, please go to our website and sign up or contact me directly if you have any questions. We look really forward to working with all of you.
Thank you, everyone.